Hi, good morning, and welcome to the ZP webinar, specifically actually for Swansea University. So we've put together this, let's say, webinar specifically for our colleagues at Swansea University. So let me just jump straight into it. So today we're talking about really ZP technologies for academia. Now in front of me, I do have some hardware. So I don't want you to think that this is one of those kind of webinars where it's all about just talk and a few slides. In front of me, I have some wearables in terms of microneedles. I also have a new style of potentiostat that also makes for what we call a minimally viable prototype. So I do have some hardware in front of me as well as also some slides as well. So I'm going to be talking about creating impact. I'm also going to be talking about using the ZP technology um, both in teaching, in research, and also in education as well. So there'll be quite a fast pace to this, and I will go forward. So one of the technologies I have in front of me is the Sensei All platform. So this is something that, in fact, there is plans to be doing a workshop on this. I've got a suspicion about October or November time actually in Swansea, and we'll do some lectures and we'll also do some practicals upon this. And also today I'll be talking about wearables a little bit. Some of you will know a gentleman called Rajiv Shah at Swansea University. He's very interested in microneedles. This is a microneedle technology that ZP has developed. It's got something like 50 patents behind it. It's had something like 20 million euros spent on it so far. And it's something that ZP does license into our client base. I'll probably mention our business model as we go along, but ZP's ambition is not really to be what we call customer facing. It's actually to be a technology provider and OEM supplier. So today I will talk about IVD in vitro diagnostics, and I will talk about wearable biosensors as well. And I'll be in particular always mindful of ZP's connection with Swansea University when I actually are talking about some of this technology. Something you may or may not be aware of is something that we call, actually we call it Julie. So there's a cloud system out there called Julie. I've put the URL in here, but if you ever want to find this, you just have to Google Zimmer and Peacock and Julie, and that's Julie with a D, and you'll find this database. Now, lots of the technology at ZP actually feeds into this. I said database, I should say sort of data management system. So I will roll it back a few times today and kind of say, at ZP, we're very interested in electrochemical sensors. What comes out of electrochemical sensors is often electrochemical data in terms of voltage or current. But those voltages and currents are generally proportional to some sort of analyte of interest. And Julie is essentially our final repository for that kind of data. So Julie is something that you can open an account with today. And some of the Sense It All. So I've got an app in front of me on my phone. We call it Sense It All. And this app will actually drive much of our hardware. And when it drives our hardware, it'll also be a bridge between the hardware and actually Julie, and all the data will get saved to the cloud. So themes today: education, research, and translation of academic research into actual products. So if I just change cameras just for a second and put my hand on here, this is the Sense It All platform. Um, I don't want to say it's a potentiostat because potentiostats kind of have a kind of, people have a certain expectation on potentiostats, but it does take screen printed electrodes. It's a very modern type of potentiostat that we can take these screen printed electrodes. We can put the SPEs in there, the screen printed electrodes in there. But it's very modern in terms of it's actually driven off my phone. So at the moment, if I want to do an amperometric experiment, amperometry, I have a QR code on my screen. So I'm just going to change. Yeah, in fact, what I'll do is I'm just going to scan that code, and I will just change. I'll just stop sharing for a second. I'm just going to share my other camera. So what I'm showing you here is that I wanted to do amperometry. And by scanning that QR code, my phone now is set up to do amperometry. And that phone will then control essentially the potentiostat. So this is a very quick way of going between what we call different electrochemical methods. So I'll just share my screen again. And we've got amperometry here. We have open circuit potentiometry here. So open circuit potentiometry is anything that's, if anyone's ever made a pH measurement in the lab, then that's generally open circuit potentiometry. Now I'll do another QR code. This is electrochemical impedance spectroscopy. So now that's impedance spectroscopy. There's quite a lot of work at Swansea. People are doing protein binding assays. You could sometimes call those immunosensors. So EIS, electrochemical impedance spectroscopy, is quite a thing, let's say, when you're doing that kind of work. We have linear sweep voltammetry, so I'll just scan that. And then the app is able to now do linear sweep voltammetry. When I say the app, see, the app is actually in Bluetooth connection with the hardware that's on my desk. So this is a very modern, let's say technology, something that is happening at Swansea. Several people are using cyclovoltammetry. Now this app is set up for cyclovoltammetry. So this app will then make Bluetooth connection with the potentiostat. The potentiostat will run the cyclovoltammetry and give the data back to the phone, and the phone will actually upload that data to the cloud. And then just finally, we've probably got square wave voltammetry, which is something that also is a methodology that is used at Swansea as well. If any of you know Fionn, then she does use square wave voltammetry at Swansea as well. With that said, I'll go forward a little bit more. Now, what's interesting about this is, so I'm showing you that we can do fundamental electrochemical measurements. And when I say fundamental electrochemical measurements, I'll also hold the potentiostat up as well. So we can do fundamental electrochemical measurements, but very quickly we can actually make the system do specific analytes. So what's holding back academic research from having impact is that the research finishes with raw signal, but raw signal is not useful to anyone at Morrison or Singleton. The clinicians are not interested in raw signal. They really just want the value. So in fact, in order to do that, the SensiAll platform is actually able to become analyte specific. So, we've done chili and ginger, we've done pH at ZP, we've done nitrate, we've done total antioxidant capacity, we've done glucose, we've done potassium. You will see it, and I haven't really emphasized it at the moment, that this app that I'm sort of waving around on my phone is already available on the Google Play Store on the Google Play Store and on the Apple App Store. So if you open an account on Julie and later on download this app onto your phone and you log in using your Julie credentials, this app and Julie are actually then synced with one another. So first of all, you open up a Julie account, so go online, find it, put your email in there, create a password, and you've got an account. Then when if you download this app onto your phone and log in with those credentials, those two things are essentially synced. Then the cloud and the app are synced. And then you're going to find it's very easy to go from like doing a square wave voltammetry immunoassay in the lab to actually giving people, a protein concentration, etc. So, and that's something that we can sort of train people to do. So this is a system that both does fundamental electrochemistry and is a system that can then become analyte specific, and you can roll it out, much easier to clinical partners than otherwise in a normal sort of, let's say, potentiostat. So I will talk about microneedles today. If any of you are aware of met Rajiv Shah, he does a lot of microneedles. If you see him around the building, do mention to him about this particular technology. So ZP does have microneedle technology. I will talk about screen printed electrodes at the moment. As I'm making this webinar specific to Swansea, I'm aware of the screen printing at Swansea, and I'll talk about reproducibility of screen printed electrodes. I'll talk about electrode customization, the pros and cons. There's a lot of cons. There's a lot of downside in customization of electrodes. I'll talk about electronics just very quickly, and I'll also talk about commercialization strategies. The government is very interested these days in demonstrating impact. And impact in some ways says, this technology, it wasn't just, you could measure impact by saying we did outreach programs, which people are very, keen on doing. But you can also demonstrate impact by actually either commercializing it or at least getting it into the hands of clinicians and doing some sort of clinical study, or even if it's just a pilot study around it. But in order to have that impact, you have to get higher than what's called a technology readiness level number 3. In development, you have a scale, technology readiness level number 1, all the way through to TRL 9. Now, most research actually gets stuck at TRL 3. But in order to really have impact and get it out the lab, you have to get it more robust, which is the higher technology readiness level. So that's something I'm going to cover today. Also, the technology can be played into undergraduate teaching and also into postgraduate research as well. I'm not going to bear on this, but ZP's been around for 10 years now. We're ISO 13485. Personal comment, I just happen to be a honorary senior lecturer at Swansea University. But one of the things I'm going to cover now is wearable biosensors. So at ZP, as I say, I've been working on wearable biosensors for at least 15 years myself. Started my sort of wearable biosensor journey probably in California when I was running and working out there, running businesses and working out there. So what I'm putting onto my wrist at the moment is basically a wearable potentiostat that's got microneedles. So if I flip this back off my wrist, we have a insert in here. This insert has microneedles on it. This insert is ran by the electronics in the watch. The watch goes onto my wrist, and by tightening up the strap, I'm effectively pulling some microneedles through my skin and into my interstitial fluid. Now, let me run through some of the configurations that we have on wearable microneedles at Swansea. We have wire sensors. So if any of you, in fact, I know one of the master students that we did a master's project with Zimmer and Peacock. They ended up at Abbott. They ended up at Abbott doing glucose strips. But Abbott also has what we call the Abbott, they call it the Abbott Freestyle Libre 2. So this is an Abbott product. This is not a ZP product, but this is a good example of what we call a transdermal CGM, continuous glucose monitor. And this wire basically goes through the skin. It has an enzyme on it, so it's an electrochemical enzyme sensor, and it's for detecting glucose. So at ZP, we do have a lot of experience on doing these kind of wire or filament sensors. We do have a lot of experience of doing microneedles. I think we've got possibly the world's largest patent portfolio on microneedles, or at least microneedles for biosensing. We do some non-invasive work, measuring things in the sweat, and we do also do some implanted sensors as well. And I'll have some slides on that as well. This is one of our implanted sensors, in fact. This is a package that actually is quite small. It's only sort of 50 millimeters across on its narrowest point, and about 38 millimeters long. Quite small, not terribly small. You wouldn't want that under your skin, but in fact, it does actually get implanted into fish. It's not something that we've done at Swansea actually, but Swansea, just FYI, I'm sure many of you know this, but Swansea does have a sort of I want to say a RAS system, a recirculating aqua system, where they actually do sort of fish studies in there. So at ZP, we're also doing fish studies. This is a RAS system in Norway. And we're actually implanted those sensors that I was just showing into these fish, and we're monitoring their glucose and cortisol in real time. So this is, can you imagine how hard it is to make it, let's say, an implanted sensor, but make an implanted biosensor that actually goes in a live animal, and the live animal is then then swimming around underneath the water, and we're getting the data out in real time. That's a real engineering challenge. That's something that we have done at Zimmer and Peacock. So this is a day study where we've got the baseline of the fish, then the fish are basically fed, and their glucose levels go up accordingly, and then they start to come down again as the fish metabolizes what it's ingested. So that is something that we've done at ZP, and we are using those kind of technologies in our biosensors. Something that I always kind of say to people is, that, I mean, at Zimmer and Peacock, we are biased, but electrochemistry as a science for gathering information about human health is a good science. It's really proven itself to be what we call scalable. You could never argue that electrochemistry hasn't proven itself to be a technology that you can produce at mass, and that you can make low-cost strips and low-cost meters, because every glucose strip out there today is an electrochemical sensor. An electrochemical sensor. You actually find people even now actually saying, "Oh, let's do an optical system." If you look at the history of glucose strip testing, they all started off optical. They only became electrochemical later on. So optical first, but then they were superseded and improved upon by electrochemical sensors. And that technology now has, translated over to these CGM sensors. So they're basically using the same science. So don't be in any doubt that electroanalytical techniques, electrochemistry, or make for good point of care sensors or for good wearable sensors. And now I think the next phase in CGM, continuous glucose monitoring, is probably going to be microneedles. It's much more acceptable. I mean, I know that Abbott, for example, are very interested in actually putting sensors onto people who are otherwise quite healthy. You'll see sponsorship where they're sponsoring athletes to wear these. So there's an interest in sort of metabolic health, but really to penetrate the market. In fact, this is the applicator for a modern CGM. It's quite a large bit of plastic. It has a large spring in it, and it sort of fires um, this sensing package onto and through the skin, which is great. But it's quite a large, amount of carbon footprint in that. So I really think that, in fact, the way that a microneedle system works, where you don't need the applicator, is going to be, for most people, in the long term, if we're going to want to measure our kind of our metabolic health, diagnose pre-diabetes and prevent pre-diabetes, then a technology that's a little less invasive with a little less carbon footprint is probably going to be the solution. And of course, at Zimmer people, we're pretty biased because we do have this technology that's had 20 million euros invested so far and has 50 patents behind it, which is really important because in the world of biosensing and biosensing in the medical space, freedom to operate, which is not insured but is supported by a large patent portfolio, is a useful thing. We do have 50 patents around that. We do have microneedles available on the web store. We do have wearable electronics also available on the web store. And we are coming out with a wearable potentiostat pretty soon. So watch this space. I am going to talk now about the Sensei All platform. This is the Sensei All platform. Like I said, I've actually got the technology in front of me as well. So this won't be just a lot of talk. I will do a demonstration shortly on this platform as well. So this is our this is our IVD platform called Sensei All. As I say, we also can use the same technology stack also for doing wearables as well. Something I want to talk about a little bit is screen printed electrodes. I know at Swansea there's a lot of people working on GFETs, graphene field effect transistors. In fact, Swansea, we do sponsor a PhD who's currently doing work on GFETs for gas sensing. So I'm not going to talk about GFETs this afternoon. I'm actually going to talk about screen printed electrodes. I think when it comes to liquid phase detection, I think that electrochemical sensors are actually not, are a good technology. I actually have evidence that GFETs are pretty good for the gas phase. But if I'm talking about liquid phase detection, that's in urine, blood, saliva, river water, sea water, any kind of liquid phase. And I think electrochemical sensors are pretty good. Electrochemical sensors have already proven themselves, commercially. We've got obviously these glucose strips, and they've also proven themselves to work perfectly well with multi-analyte detection as well. So this is a blood gas analyzer from Radiometer, which is also electrochemical and is using thick film printed biosensors. So screen printing or thick film printing has already proven itself to be A, it works, and B, you can make commercial products on it. Just as a quick comment as I go through this slide, this particular slide is of the One Touch Ultra, which was made by Johnson & Johnson a while back. And it's actually the best selling, or was the best selling glucose strip on the market. And I think it's something that's, I was talking about technology readiness levels earlier on. So as you go up these technology readiness levels, you've actually got to make your technology usable by normal people and acceptable by normal people. Now, this is actually a product, and it was accepted by normal people because actually it was the best-selling product on the market. Did that acceptability come from best science and best engineering? It came about because actually it was available in multiple colors, and people could change the casing themselves and actually choose whether they were going to have a hot pink or a sort of neon blue or maybe just a metallic finish. And there were more colors than this. So very quickly, I'm just saying that as you come up the technology readiness levels, you've got to make it appealing to real people, let's say, and how One Touch, which was made by Johnson Johnson in those days, was actually made appealing was by making it available in many colors. So sometimes it's not the science that matters, it's the psychological um, factors, let's say, that can make something successful. Now, at Zimmer Peacock, we do supply screen printed electrodes into some groups at Swansea. I think the main problem with screen printed electrodes in general is this, that people have an expectation on a low price. So at ZP, we do provide these electrodes at a low price. The particular screenshot there, that's like €1.76 each, but we do another version that's like 90 cents each. 90 cents is about 84p, so we can make the price. And many other people do a low price as well. But the problem is that I think at ZP, I'm super biased. You've got a, disclosure of interest. As an owner of this company, I am biased. I think we make a really good quality of screen printed electrodes. In fact, we were feeding some of our gold electrodes into a undergraduate project, and I know that they were quite at Swansea, and they were quite surprised. Well, not surprised. They shouldn't be surprised, but they were quite pleased how good those gold electrodes were. So we do a good price and we do a good quality. There's a general problem in the overall screen printed electrode commercial market, and that problem is there's more choice and demand, and there's also an oversupply and customization. So I'll talk about people want a low price, they want high quality. There's a problem. There's actually a limited demand in the market, and there's so many suppliers. It actually leads to an oversupply, and there's also a willingness to do contract printing for people, which also then leads to a problem in quality. So I do want to define that. So this is, maybe I want to change my analogy one day, but unfortunately in the military, if you're making munitions or you're making something that, body armor for soldiers, you want a high quality, and they're very willing to pay a high price for it. So unfortunately, defense spending, which they want to bring to 2.5% of the sort of GDP, the gross domestic product of the UK, they want to bring that to 2.5%. The military has to spend a lot of money on equipment because they need it to work every time. So they want basically a high quality, and they're willing to pay a high price. Now, if we look at academia, then people, and I'm talking about screen printed electrodes in particular, they want a high quality, but they are unable to pay a high price. So in fact, they want the quality, but they actually want the price lower. So they're not like the military, which will pay a high price for high quality. Academia is different. They want a high quality, but they want a low price. Now, the only is a strong term. The only way you can make a sustainable, high quality, low price product is actually have high volume manufacturing. So, for example, these Android phones, you can get a good Android phone for less than £200. Why is that? That's ridiculous in some ways. Well, the reason that you can is because actually of the high volume manufacturing. So in order to have high quality and a low price, you need high volume manufacturing. But I'm going to talk about the high volume manufacturing at the moment. So let's talk about screen printed electrodes. Now, the reason that you should care about screen printed electrodes in some ways is because I'm just grabbing a screen printed electrode off my desk now. So I hold the screen printed electrode off and say, these kinds of technologies have proven themselves to be very good at translating research into real world products. If you were wondering how, I'll hold up a real glucose strip, let's say. This is a glucose strip, and it is made by screen printing. So the glucose strip has demonstrated to us that screen printed electrodes are a commercially viable technology for manufacturing biosensors. The problem that we have is, and ZP is part of that problem, so ZP is one of many. There are many people on the market screen printing electrodes. If you ever, if you've been doing this for as long as some of us have been doing it, you find that some of these people come and go. But there's always quite a few people on the market any one time willing to do screen printing. And you'll also find that they actually have a large portfolio of products, which sounds great. Lots of vendors producing lots of products. And then you will also have, academic groups also screen printing either for themselves or screen printing for other colleagues. So there's a whole mix of people making screen printed electrodes. But let's say there were 10 vendors in this world, When I've been a vendor, Zimmer Peacock's a vendor. There are other people making electrodes for academia, and so academia can do its biosensor development. So let's say there were just 10 of them. In fact, there is more than 10. There's probably more like 20, 30, maybe even 40 out there. There's a lot. Let's just say there were 10. And let's just say that 10 all had a catalogue of 50 sensors. I'd actually think that actually many of these 10 actually have more than 50 sensors. But let's just say on average, every vendor was producing 50 sensors. So that means that there are 500 or more products on the market. So you're an academic, you want to make a biosensor, you think you need some screen printed electrodes. You have a choice of more than 10 vendors, probably 50 products per vendor. That's 500 products available to you. It sounds great, it sounds absolutely great. But this is the problem. How many tests or experiments per year on a screen printed electrode do you think are taking place in academia? I would suggest it's not more than 1 million. Could you imagine, you think about, if you're doing GFAT work or you are using screen printed electrodes, etc., and you're at Swansea, think of your colleagues, think of yourself, how many experiments are you really doing? And then scale it for all the other universities around the world. Is it really more than 1 million experiments happening per annum? So let run with me on this. Let's say there are 1 million experiments per year. That's 1 million screen printed electrodes. But 1 million screen printed electrodes, but there's actually more than 500 products. So if I take 1 million and divide by 500, that means that any one product is only selling about 2,000 per year. So that means, and if I take 2,000 per year, I'll just hold up this sheet. This sheet has 300 electrodes on it. That means that in order to fulfill the entire global demand for a particular product, I only need 7 sheets like this. 7 sheets like this, it's hardly worth turning the screen printer on for that. We could produce that, in some minutes. At least a print like that would just take a few seconds. So what am I saying here? I'm saying there's a lot of vendors on the market. Great. They've got a lot of products. That also sounds great as well. But actually, there's only probably about 1 million R&D experiments per annum happening on screen printed electrodes. So that means that really each one of those products is only being sold in something like 2,000 per annum. 2,000 per annum is printing 7 sheets. I would argue, and I know I'm getting into manufacturing here, but I said that to have high quality and low price, you need high volume manufacturing. But that calculation there tells you that, in fact, none of these vendors are really doing high volume manufacturing. I wouldn't even call it low volume manufacturing. It's almost nothing sort of manufacturing. So a lot of these SPEs are produced in very low volumes. And if they're producing higher volumes, then they're basically sitting on the shelves for years. And this is materials. So materials are not just inert. Materials do change over time. Gold tarnishes, platinum oxidizes a little bit. Carbon will also oxidize a little bit over time. So you're finding that, in fact, what will happen is somebody will produce a batch of electrodes, and actually they just sit on the shelf for years. And they're aging over that time. It's no criticism on the manufacturer. This is I think the only forum where you'll find people talking about the reality of what's actually happening here. Now at Zimmer Peacock, we're trying to change that model. So we understand that actually people expect a low price, but actually they're getting a low quality for it. And anyone who's using screen printed electrodes knows that actually there is a problem with quality. Because actually they're getting a low quality at a fairly low price. But in fact, at Zimmer Peacock, we're trying to keep the price the same, but actually trying to give a higher quality. And how do you do it? You do it by actually doing high volume manufacturing. So at Zimmer Peacock, we're as guilty as everyone else that, we've got a large range, let's say, of screen printed electrodes. But in fact, most of the time now, we're actually saying to people, if you're doing a carbon electrode, take our 501 carbons. If you're doing a platinum, take our 501 platinum. If you're really on a really low cost carbon, take our hypervalued carbon electrode. That one's 90 cents, which is about 84p, I think. If you want a low carbon, a low cost platinum, take one of these. If you want a low cost gold, take one of these. And we are actually shipping some golds to a group in Swansea very shortly, and there are actually some golds in action as well at Swansea at this moment, which have had a really good feedback, and they'll probably find themselves in a publication pretty soon as well. So at ZP, rather than, what we're doing is saying, okay, we do have a large portfolio of screen printed electrodes, but it doesn't do you any favors to have all this choice, because all it means is that we're not really manufacturing anything in high volume. So we're very, strong with it and say, look, if you want a gold electrode, this is the gold electrode. If you want a carbon electrode, you've got a choice of two. If you want a platinum electrode, you've got a choice of two. Not because we're trying to be, restrictive, but the reality is, the more people that use these electrodes, the higher the volume, the price stays low, and the quality gets high. And we're really good on the quality. So, for example, these are now €1.76 each, but these are 90 cents each. This quality of screen printed electrodes is important because I think it's what's really holding back a lot of academic research. I want to talk about that in a minute as well. So let's talk about quality. So when it comes to quality of screen printed electrodes, I think people are using the wrong metrics. Now, maybe the workshop that we'll hold in Swansea will help with this. But when I say the wrong metrics, people are very interested in looking at what's called cyclic voltammetry on screen printed electrodes, and they like to look at the peak current versus square root of scan rate. Now, that actually doesn't tell you so much about the quality of the electrode. It actually just tells you about the diffusion control of molecules to the surface. The other metric that people are quite interested in is actually peak-to-peak separation. So if I take a molecule like ferricyanide and I reduce it and I oxidize it, I'll get a cyclic voltammogram, and the peak-to-peak separation between the oxidation peak and the reduction peak, theoretically for ferricyanide, because it's a one electron process, should be 59 millivolts. With ZP, we get, for an example, 67.5 millivolts, which is good enough as far as I'm concerned. But what I find is that people are very, very interested in, your peak-to-peak separation. The peak-to-peak separation shows you that you have what's called Nernstian behavior, and that's great, but it doesn't get you to market. It doesn't give you impact. I think we're using the wrong metric. The metric that we should actually be interested in is reproducibility. If you come to one of our, ZP has a product line called Food Sense, Google Food Sense, Google Food Sense and webinars. Hopefully you'll be able to find our pages. If you come to one of our webinars, you're going to see that when we're doing measurement of food ingredients, we do no calibration. Now, when you read academic papers, they're often normalizing the data. I'm even thinking about GFET data. When you look at GFET data, they often show it as a percentage change. What they're effectively doing is a single point calibration, and because from sensor to sensor, there's actually quite a lot of variation. But you will find out with ZP that we don't do any calibration and we don't do any normalization of data, which leads to simple assays. Simple assays actually lead to easier to commercialize. And if you can commercialize something, then you're more likely to be impactful. So that whole, let's say, discussion I just made on screen printed electrodes was just to come down to the fact that we're trying to give high quality at a low price. But by giving high quality, it suddenly removes one of the problems that is otherwise facing people in the biosensor development world, which is actually sensor to sensor variation. And because of sensor to sensor variation, they end up trying to do calibration on their devices in the field. And if you do calibration on the device in the field, then you have to have more complex microfluidics. You're starting to have calibration solutions. None of this is going to translate well, and it's certainly not going to lead to a low cost device. So at ZP, we focus more on reproducibility of screen printed electrode. Though we can get a peak-to-peak separation close to 59 millivolts for a simple redox system like ferrocyanide, though we can get close to that, we're more interested in actually the repeatability of our screen printed electrodes, because I should say reproducibility. The reproducibility ends up allowing us to take screen printed electrodes and make products from it much quicker, or make assays from it. So in fact, when we're producing, we're actually doing a lot of electrochemical testing to make sure that what we ship, just grab it now, we'll take a sheet like this. We'll take several samples from each sheet and actually make sure that essentially the cyclophotometry in this case is actually repeatable, so that when you build a sensor upon it, you get a signal that is, for example, proportional to concentration, and not a signal that is influenced by concentration and by the electrode itself. So we're trying to make your signal only proportional to concentration and not be dependent on the electrode that you happen to choose at that time. So we're very careful about that. So we will look at the peak heights, we will look at the standard deviation, we'll try and make sure that our relative standard deviation is single digits. So this is, we're looking at a production line where we're looking at like 21 sheets of these, so we may produce, 21 to like 100 sheets, and then we'll test within that production batch to make sure that our repeatability or our relative standard deviation is 6%, in the next case 5%, in the next case 6%, in the next case 6%. So you can understand that the underlying, let's say, repeatability is good enough that in fact you can get a signal out that's proportional to concentration, and not just proportional to concentration and some offset that's due to the irreproducibility of the underlying electrodes. So I think that people are using the wrong metric. They shouldn't be thinking about Nernstian behavior on a screen printed electrode. They should be thinking about repeatability of those electrodes. So when ZP then does, we will test a bunch of screen printed electrodes with different concentrations. Each color represents a concentration, and you'll find out that actually we have very good, even though there's 3 electrodes for each one of these, let's say, colors, they're very tight, or they have a very small error bar associated with them. And that again leads to simple assays. Simple assays leads to commercialization. Commercialization is part of being impactful. Just want to say, if you are using screen printed electrodes from ZP, make sure you have good connectors. In fact, at Swansea they do have another potential stack from another operator, but they do have the connector from ZP as well, because otherwise, if you have poor connections, even our screen printed electrodes are going to look poor. So make sure you do get the right connectors if you are going to use our screen printed electrodes. People do ask for customization. This happens too much in, that people will have a very, I need a specific design. I can do lectures specifically on the idea of using a working electrode and using what's called a null electrode and doing a differential on it, but it's quite a common request. We'll get requests about doing arrays of electrodes. This is also an array of electrodes. So these are kind of, sorts of requests. People say, I need a very bespoke electrode for my application. But what people and interdigitated electrodes is also something that's requested quite a bit as well. The problem with bespoke or custom electrodes is you will find many vendors, sure, they'll do it for you, but it comes back to the thing that, in fact, what you're going to end up with is a low quality product. Why? Because it's actually low volume manufacturing. If you have something custom made for you, how many sensors do you, or how many screen printed electrodes do you really need in 1 year? Possibly 1,000. 1,000 is 3 sheets of this. That's not manufacturing. So in fact, there's a problem that people think that they need customization. They can get customization done. They want it for a low cost. So if you want it for a low cost, sure, people will do it for you for a low cost. But in fact, that low cost also means that you also, because especially in the academic setting, you don't really have the manpower to do high volume. So in fact, you need low volume manufacturing. Low cost times low volume equals low quality. Now, in this particular case, low quality can mean low yield. So this particular company, they're now in liquidation, or so they went bankrupt. And in part, it's because, in fact, they wanted low cost when they wanted this. They didn't come to ZP for this. ZP was apparently too expensive. But they ended up paying for their low quality because they had to do a lot of what's called IQC, incoming quality control. And in fact, what happened is they found that only a third of what they received was actually any good for them. So they ended up paying for that price through extra effort in order to make sure the electrodes that they were putting into their product were actually any good. And so they end up having to have people in the team who are only testing electrodes. The big secret about these electrode geometries that you need is at ZP, we've done a lot of electrode geometries, a lot of materials. And you know what? Often, this is a gold electrode, this is a gold electrode, this is a platinum electrode. We were using these very different configurations to measure conductivity. Different materials, different configurations. horrible answer is it all gives the same results. So people are very obsessed that actually, what's the electrogeometry I really need? When you're talking about biosensors, you're talking about low currents, low volumes of material, and you're talking about small gap distances between the electrodes. In that scenario, your electrogeometry is actually not that important. So you don't need customization the same way you want. I mean, we appreciate that most people actually in academia are actually pipetting. So we made a nice circular electrode to kind of help people easily pipette onto those electrodes. I think that, and directing this specifically at Swansea, your value add is actually not necessarily in printing electrodes. It's not necessarily in customizing electrodes. It's actually in coming up with that electrochemical assay. So if you're making an immunosensor, developing that biochemistry to detect that molecule is the real value add. How are you going to get the signal out of that assay is the real value add. So I put square wave voltammetry up here. It's quite a sensitive technique. It's quite good. I'm sort of thinking in particular about immunoassays. Square wave voltammetry is pretty good. In fact, when I think about Swansea in particular, we've done some testing of THC, which is tetrahydrocannabinoid, and CBD, which I forget, but it's a sort of it's not an analog, but it's similar to THC. And there we were using, I think it might be differential pulse voltammetry. So I'm saying the value add in your academic research is the assay itself. That's the bit that is patentable. The methodology for making that sensor work, that is a value add. The sorry, I labeled it here, the calibration, how to get the raw signal into a concentration for the clinician, for example, that's a value add. So if I said there are three things you should be doing in your research: developing the assay, developing the method to actually get that assay to work, and then the calibration, which is really a way of getting the information out of the raw signal. Now at ZP, we've built an entire platform that means that you can focus on these three things and not get distracted by everything else that's, otherwise not value add. There are lots of people who are trying to develop electronics in the world. I do have a bit of an opinion on electronics, that actually electronics for biosensors has become very commoditized. In fact, we're talking to a large multinational semiconductor company this afternoon, and there they've got multiple products that can be used in biosensors. So even a single company electronics, when I say electronics, I should say semiconductor companies these days have multiple products. So there's not really a value in developing new electronics, I would suggest, for biosensors. If you want electronics, they're off the shelf. You can definitely get them from Zimmer Peacock. Our electronics will do amperometry, voltammetry, potentiometry, and impedance spectroscopy. We can do wearable format. We can do IVD format. That means in vitro diagnostics format. We will list open source potentiostats on the website, but I do think the real value in open source potentiostats is training electronic engineers, and that's an important, let's say, effort at academia. But don't think that the outcome of that open source potentiostat is a value in itself. It's the developing it and training that engineer, and that training and that engineer going on to join industry. That's the real value. It's not the electronics itself. So, because I think one of the things is that what we're trying to focus on is that user experience. Actually, when I say user experience, can you think of how many instruments that you use in the lab that you could really give to people? Let's say a clinician at Morrison Hospital who's very interested in measuring the pH of blood. How many of those instruments that you're testing upon or using could you really go and give to that clinician? And the answer is none. But what I'm going to show you this afternoon, actually, it is translatable to products that you could otherwise give to clinicians, for example. I mean, when I say this, these are all things that ZP has done. We've done benchtop instruments, handheld instruments for detecting on the breath. So we are very used to the user experience. That's just to give me a little bit of credibility when I make the comment that you have to think about that user experience a little bit. This is something that when I left my PhD, this was already something that was known. It's called the Valley of Death. So the Valley of Death describes this, that you've got technology readiness level number 1, 2, 3, 4, 5, 6, 7, 8, and 9. When you're at technology readiness level number 9, you're at your most robust. You're really a product. But most academic research actually finishes at technology readiness level number 3. So, I mean, I'm aware of, at Swansea, multi-channel or parallel channel large potentiostats. Those potentiostats will leave you at technology readiness level number 3 because you can never translate that instrument immediately into a hospital setting. It's too big, it's too bulky, it's too complex. Um, Governments, um, including the Welsh government, spend a lot of efforts trying to get technologies out of universities and into the commercial setting. And the gap between where academia finishes and where companies can take over is called the valley of death. That most technologies actually just fall into it and never span this gap. So governments try to span the gap by you've got organisations like Innovate UK in the UK, also KTNs, knowledge transfer networks, and KTPs, and knowledge transfer partnerships. These are all designed to try and get technologies out of academia into industry in order that essentially the government wants more jobs, more revenue. So at ZP we say stop using technologies that are purely research tools. And of course I'm biased, so I've got this potentiostat in front of me. I will do a little demonstration on it in a minute. This is both an R&D tool and it's also a commercialization tool as well. And we're saying, okay, let's not actually use tools or potentiostats in our labs that are purely R&D, and I'm biased. Let's use a potentiostat that actually looks like a product. And obviously, we call that the Sensei All. This is the Sensei All. It uses screen printed electrodes from ZP, though you can use screen printed electrodes by other manufacturers. We do have design rules on our website, so if somebody wants to make their own screen printed electrodes, they're welcome to do it. It's app driven, so which is really kind of also if you're going to give products to people these days, they've got to be kind of cloud enabled. Android devices like this, they're handheld, they're easy to use. You don't want laptops kicking around. As I say, it's cloud enabled as well. So that all exists, and I'll give a little bit of a demonstration on that shortly. You can scan this QR code. It's hard to do whilst I'm doing a live webinar, I admit, but that QR code will show you that that app actually exists on the Google Play Store. You can scan this from an iPhone, and that will show you that this app exists on the Apple App Store. And we're very keen to actually get this into undergraduate training because we think that this potentiostat is relevant to undergraduates. It shows an up-to-date, mindset, and it can be used in lab practicals. It's such a simple instrument. I'm sure if you're using GFETs, you're used to probe stations. If you're using screen printed electrodes, you're probably using slots or crocodile clips. This is how quickly or easy it is to set up a sensor. Take the sensor, put it in here, close that, electrical contact made, done. So it's very good for doing both research and lab practicals, but also undergraduate lab practicals as well. So we're good for education, we're good for research, we're good for impact, and I will make that whole journey, let's say. So I actually, this is a sort of 10-year journey for us, but you could appreciate that at ZP that, we're very keen on actually starting now with the undergraduates. The undergraduates become the researchers, and the researchers become the entrepreneurs of the future. And one platform can go all the way. So we want to replace this mindset of using horrible cabling and large potentiostats and even laptops with actually instruments that are much more relevant, up to date, and easy to use. And I would say that the Sensei All is actually meeting all of those criteria. As I say, you can actually find these apps on the web store. And when you have got that app on the web store, at the moment, my phone says square wave voltammetry. I'll just scan this QR code. Now it says amperometry. But now I can actually run my Sensei All potentiostat as an amperometer. If I scan this code, I can do open circuit potentiometry. If I scan this one, EIS, this one linear sweep, this one cycle voltammetry, and this one square wave voltammetry. So it's I'm just going to scan this code for a second, actually. Well, this is glucose, so let me just scan glucose. App has just become a glucose sensor. You can actually be part of that. You can develop an assay using one of those methodologies, and then you can actually translate it to actually not show you the raw signal, but actually just show you the concentration of the analyte that you're particularly interested in. So I'll just do chili, for example. And then it says chili. Chili always makes me smile. I think we'll be doing a workshop in Swansea on that in, as I say, either October or November, I got a suspicion. But I'm just demonstrating to you that this QR code driven technology means that you can go from lab experiments to analytes really quite quickly. And we've built into it, this is a QR code here. If you scan this QR code when you're watching a recording, it's going to tell you that in fact you can have an interface where you can edit all of this. So I know this is a commercial comment, but OEM means original equipment manufacturer. ZP sees themselves as an OEM provider, which means that other people can build technologies upon our platform and they can go to market with it. In order for that to happen, you've got to be able to have apps out there driving your assays that don't say Zimmer and Peacock. And in fact, we've built it so that in fact you can edit all of these fields and all these colors. And in fact, the app will say, Swansea University, for example. Now we are trying to place these actually into the undergraduate teaching. So if you're aware of who's doing undergraduate teaching at Swansea University, especially on electrochemical analytes assays, please let us know, because we think this is a good place to start. But what I want to do now is I want to do a quick demonstration, actually, just demonstrating that we can make a very quick measurement. We can actually get that data into the cloud. And what's really cool about this is it will give me a result on my phone, but it will also put the raw data into the cloud as well. So I'm just going to stop sharing for a second. I'm going to change cameras. Yeah, so you can see that. What I'm just going to do is, this is the new type of potentiostat. So here's a screen printed electrode. I'll just take a sample and I'm just going to pipette 50 microliters. This is the wonderful thing about electrochemistry. Unlike optical systems, it really requires no sample, relatively no sample. So I'm just going to actually, to make electrical contact with that screen printed electrode, actually, I just need to click that, put the sample on like that. And now I will say, just going to give it a file name, because what's going to happen is this data is going to go up to the cloud. I was talking about user experience earlier on. As an electrochemist, and also a guy with some background in GFET, nobody really likes our raw signal. So when you're really thinking about real end users do not want to see a raw signal. What I do want to share with you is, in fact, let me just bring something up real quick. Yeah, here it goes. So I'm going to share my screen real quick. And what I'm going to do is I'm going to log on to something called Julie. So let me just log in here. So this is a cloud system where ZP, I want to say sort of ZP puts most of its data. So whenever we run an assay, for example, in fact, here's Swansea here. Yeah. So every time we run an assay, in fact, what is happening is that the data is actually going to the cloud. So for example, if I do a test, then the cycle of voltammetry is ended up in the cloud. But there's very, very powerful tool here. So my name, so I'm at Swansea, I'm doing a PhD, I might be using square wave voltammetry, for example, and I may be gathering, now this is cycle voltammetry, admittedly, and I might be gathering data like this. Now, that is no good to a clinician. It's no good. But in fact, what we've done here is we converted it into concentration, micromolars in this case, and we've sent the data back to the phone. But how did we process the data? We've got we've got all these tools that say this is scan 1 and this is scan 2. On scan 2, we've done what's called filtering. So whenever you gather data in the real world, it's going to be noisier than what you've gathered in the lab. And then we've actually told the software, look for a peak in this window, and when you find that peak, convert that peak into concentration using this equation. You can actually see what it's done here, that we've found the peak and we found the peak height and we've turned the peak height into concentration, and you can just program that in the cloud, and that raw signal will then go back to the phone. So the clinician, like I say, at Swansea or Singleton, actually gets this result of concentration and doesn't get your raw signal. But if you're trying to troubleshoot this, it's really useful. I'll show you some, some sort of stupid signals. Let's say this blank here. If something went wrong, and it looked like that, you would know about it in real time. And so you don't have clinicians kind of complaining to you that it's not working, because you could actually say, I don't think you put a sample on the sensor. You can troubleshoot it quite quickly. So I'm going to do a quick summary now, and I sort of appreciate that. If you're so interested, there is a workshop being organized. We're going to have a meeting about it next week, and it'll be happening in Swansea, and I think about October and November. But microneedles, if any of you run into Rajiv Shah, just mention to him that we did get a shout out to him during this webinar. And like Rajiv, we are very interested in microneedles. We are very hopeful that more people adopt our screen printed electrodes. They're really reproducible. They will really help you make much more progress on the assays you're otherwise trying to develop. I have talked about the pitfalls of screen printed electrodes and the pros and cons of customization. In fact, I've said there's no pros. There's no pros on doing customization when you're actually at a very low volume. Electronics, you've seen that, we've got a very powerful set of electronics. And then that commercialization, in fact, I can go from potentiostat to customer facing product just with the use of a QR code. So we very much hope these things get adopted. So I will say I've been speaking for 56 minutes. The most valuable resource you have in the world is your time. So I don't want to take up any more of your time, but if you've got any questions, as always, don't hesitate to reach out to us, and some of you will see us on campus pretty soon. Okay, thanks very much.